Hi everyone, my name is Mariola Gres Capdeville. I'm a research associate at the Department of Animal and Dairy Sciences at the University of Wisconsin-Madison. I work with Dr. Thomas Crenshaw, and today I will present some of the results from our studies that were designed to evaluate dietary phosphorus adequacy for sows. So I will start this presentation providing some background information on phosphorus requirements of reproducing sows, and then I will present the results from our study that was designed to estimate phosphorus requirements of sows during gestation and lactation using 24-hour urinary phosphorus excretion, and then I will discuss some practical applications from our study where we use calcium to phosphorus ratio in spot urine samples to predict phosphorus intake adequacy in sows. So phosphorus is an essential nutrient. It is involved in multiple physiological functions, and these functions include bone mineralization, energy transfer and storage, cell membrane structure, and acid-base balance. So adequate phosphorus nutrition of sows is critical for optimal growth and development of the offspring during gestation and lactation, and it's also very important for maternal health and productive performance. However, we have limited information on phosphorus requirements for sows. So this table shows the number of references that have been cited for calcium and phosphorus requirements for reproducing sows in the NRCA publications. So the NRC is one of the most important dietary guidelines that we have to formulate diets for pigs. But if we look at this table, there has been only a few studies conducted to estimate phosphorus requirements for sows. Also, since these early studies were published, there has been a large improvement in reproductive and productive performance of sows. So because of this, because we have limited information on phosphorus requirements, and there has been a large improvement in performance, diets for sows are usually formulated with excessive amount of phosphorus. So this means that phosphorus is over-supplemented. The problem with this is that all the phosphorus that is consumed by the animal but is not retained in the body is going to be excreted in feces and urine. So this has generated a lot of environmental concerns that are related with water eutrophication. So we need to know, or we need to have precise information about phosphorus requirements in order to formulate nutritionally adequate diets, but also to minimize the environmental impact that is associated with phosphorus oversupplementation. So why do we have only a few studies conducted to evaluate phosphorus requirements of sows compared with all the information that we have for growing pigs? And this is mainly because of the methods or techniques that are used to estimate phosphorus requirements in younger animals. So in these studies, the researchers usually use metabolic rates to conduct balance studies, or they need to collect bones or use imaging technologies as DEXA scans to estimate bone mineralization or bone mineral density. And because of the economic value of the sows, the maternal care of the offspring, and the large body size, these studies are usually very difficult or sometimes impossible to conduct in sows. So because of that, we decided to estimate phosphorus requirements of sows using urinary phosphorus excretion as an alternative method. And we decided to use urinary phosphorus excretion because kidneys are key organs involved in the regulation of phosphorus homeostasis. And this is through changes in the amount of phosphorus excreted in urine according to the physiological needs. So this graph shows results from a study with growing pigs that were fed a phosphorus deficient diet at the beginning of the experiment, and the amount of phosphorus in the diet was increased with days on experiment. So the researchers observed that fecal phosphorus excretion continuously increased as animals consume more phosphorus in the diet. However, renal phosphorus excretion or urinary phosphorus excretion was very low and constant until animals met the requirements. So in our experiment with sows, we used this transition point to estimate the requirements during gestation and lactation. In our study, we used 36 sows that were from parity 3 to parity 7 that were fed 6 different dietary total phosphorus concentrations. The concentration of total phosphorus in the diet ranged from 0.4 to 0.8. We also increased concentrations of calcium in the diet with phosphorus because we wanted to keep calcium to total phosphorus ratio constant across diets. We collected 24-hour urine samples that were analyzed for calcium and phosphorus concentrations. We also collected blood samples that were analyzed for different parameters, and we collected information for sow performance parameters and for offspring performance parameters. But today, I'll just show the results for phosphorus and calcium concentrations in the 24-hour urine samples because the responses that we use to estimate phosphorus requirements during these phases of gestation and lactation were based on urinary phosphorus excretion. So in these experiments, all sows were fed with the experimental diets from breeding until the end of the lactation period, and then after that, all sows received the same standard diets. We collected samples in mid-gestation or day 77, late gestation, day 112, and then in 2 days during lactation at day 4 and day 18. These two pictures show how we collected samples during gestation and during lactation. In these two phases, we used bladder catheters to collect the 24-hour urine samples. So then using statistical analysis, we model the dietary phosphorus intake, urinary phosphorus excretion curves during mid-gestation, late gestation, early lactation, and late lactation. And from these curves, we estimate the requirements as this transition point, as I explained before. So based on the estimated requirements, our recommendations for total phosphorus requirements during gestation is around 10 grams of total phosphorus per day. And during lactation, our estimate was around 36 grams of total phosphorus per day. So we know that we can use 24-hour urine excretion to estimate phosphorus requirements from sows. And the 24-hour urine collection is the gold standard to analyze solute excretion in urine because it takes into account fluctuations of urine concentration that occur within a day. However, when we are collecting samples from a large number of animals, from large-scale studies or in swine operations, a single void urine specimens or spot urine samples is a more convenient and practical method. But spot urine samples need to be adjusted for urine dilutions, and these require additional measurements in the urine samples. But if we look again at the results from our experiment, what we saw was an opposite response in urinary phosphorus and calcium excretion according to the dietary total phosphorus level. So what we saw is that animals excreted more phosphorus in urine, which is represented by the blue bars, as they consume more phosphorus in the diet, but the amount of calcium excreted in urine, represented by the gray bars, decreased as animals consume more phosphorus in the diet. So the response of phosphorus and calcium excretion was opposite in response to dietary phosphorus intake. So these results show the the potential of using urinary calcium to phosphorus ratio in urine samples in order to predict phosphorus intake adequacy in sows. So based on this, we decided to evaluate calcium to phosphorus ratio in spot urine samples as a practical method because it doesn't require the need for adjustment for urine dilutions. So this could be a practical method to monitor phosphorus intake adequacy in sow herds. So this is the same experimental timeline that I showed before. What I didn't mention is that at day 112 of gestation and at day 18 of lactation, we collected spot urine samples in parallel to the 24-hour collections. So these spot urine samples were collected from the catheter tube at 7:30 and 10:30 a.m. and at 4 p.m. These samples were also analyzed for calcium and phosphorus concentrations, and we calculated calcium to phosphorus ratio in these samples. So we used the 24-hour urine samples to calculate calcium to phosphorus ratio and to estimate urinary calcium to phosphorus ratio cutoff value to predict phosphorus intake adequacy. So in these graphs, we can see 24-hour urine calcium to phosphorus ratio according to the different dietary levels. And we saw that sows that were fed with concentrations of dietary phosphorus below the estimated requirements had a higher calcium to phosphorus ratio than sows that were fed at or above the requirements. And we saw the same pattern during gestation and lactation. So then using statistical analysis, we were able to estimate urine calcium to phosphorus ratio cutoff value for predicting phosphorus intake. And using this, we created different categories of phosphorus intake. So a urine calcium to phosphorus ratio value above 1.5 was associated with a phosphorus deficient diet. A value between 0.5 to 1.5 was associated with adequate phosphorus intake. And then a value below 0.5 was associated with excessive dietary phosphorus. Then for each of these categories, we calculated urinary phosphorus excretion as a percentage of total phosphorus intake. And as expected, what we found is that animals that were identified as fed excessive amount of phosphorus in the diet, they excreted more phosphorus in urine compared with animals that were classified as fed a phosphorus deficient diet using urinary calcium to phosphorus ratio. But these categories were created using 24-hour phosphorus and calcium excretion. But the objective of this study was to evaluate the use of urinary calcium to phosphorus ratio in spot urine sample as a practical application to predict phosphorus intake adequacy in sow herds. So then after creating these categories, we analyzed the association between calcium to phosphorus ratio in 24-hour and spot urine samples. And using statistical analysis, what we found is that regardless the physiological stage of the sows, so this is gestation or lactation, or the time at which spot samples were collected, urinary calcium to phosphorus ratio in spot urine samples explain most of the variability in 24-hour urinary calcium to phosphorus ratio. So this means that we could collect a spot sample, analyze calcium to phosphorus ratio, and we would categorize this sow based on urinary phosphorus excretion as fed phosphorus deficient, adequate, or excessive phosphorus in the diet. So then we analyzed the agreement between the two methods, between 24-hour and spot urine samples, in identifying urine samples or categorizing urine samples as deficient, adequate, or excessive phosphorus. And what we found is that a similar amount or percentage of samples were assigned to these different categories using 24-hour or using spot urine samples. So after presenting all this data that was based in urine sample collection, you might be thinking about how to collect spot urine samples or urine sample from sows. So Dr. Megan Nickel and other researchers developed some methods to collect urine samples from pigs. So one of these methods that they use with sows required a plastic bag that was positioned around the vulva, and then after urination, this urine samples was transferred into a urine cup for posterior analysis of the urine. So we haven't tried this method yet because in our experiment, we used bladder catheters to collect urine samples, but we use a similar method to collect samples from barrows. So these pictures is from one of our studies where we successfully collected urine samples applying these methods. So if you are interested on knowing more or having more information about how to collect urine samples from pigs, you should review this reference because we were able to apply the methods that they developed to collect urine samples in our studies. So from this presentation, we know that we require information on phosphorus requirements for sows because this is very important to formulate nutritionally adequate diets, but also to minimize the environmental pollution that is associated with phosphorus overfeeding. Also, urinary phosphorus excretion represents a very sensitive response variable to the level of phosphorus intake, and we were able to use 24-hour urinary phosphorus excretion to estimate phosphorus requirements during gestation and lactation. Also, the measurement of calcium to phosphorus ratio in spot urine samples may be considered as a practical method to monitor phosphorus intake adequacy in sows, and this has practical applications for mass screening of phosphorus intake adequacy in specific herds. So I would like to thank the Swine Research and Teaching Center, the UW Madison feed mill managers, and the students that helped me with collecting samples and analyzing samples at the lab. And also, I would like to thank DSM and PIC for providing financial support for these studies. If you want to know more information about these studies or you have any questions, I have left my contact information here, so please don't hesitate to contact me. So thank you for attending this presentation.